All right, so hopefully you were able to find the recordings. They're all under Unit 1 in the Lesson Units tab. So because Google Classroom turned out to be confusing for some people, I just went ahead and decided to create units within the Blackboard. Although I think Google Classroom is a little cleaner looking, but we'll just go ahead and put everything in the Blackboard. So for every unit, you will have everything that you need for that unit in that folder. So you'll be able to find your LearnSmarts there, any virtual lab that would be needed for that unit, anything that you need would be in that that area. And so that's that for the recordings. And so the recordings are there as well. And I move them up to the top for you. Okay, hold on. We have 2 more people that just came. Okay. And, yep. All right. So we're going to start talking about blood, chapter 18. And chapter 18 is our first chapter of the cardiovascular system. So you'll see, kind of like if you remember from AMP1, how nervous system was divided into several different sections. Here we have the cardiovascular system is divided into 3 chapters. So blood is our first one, then chapter 19 will be heart, and then chapter 20 will be the vessels. So blood is a fluid connective tissue, and normally when you think about blood, you think about blood transferring or transporting really oxygen and nutrients throughout your body. However, that is not the only function. So that is a big part, that is a big function of blood, but it's not the only function of blood that we see. It also transports hormones, as we just learned in chapter 17, right? Hormones travel through blood. But it also transports wastes and other things, various proteins and such. The blood is the whole blood is composed of 2 main components. Okay, so it's made out of 2 main parts: plasma, which is the fluid part of the blood, and then formed elements, which is anything that's not fluid. Okay, so this will be your the formed elements are the cells. Okay, so we're talking about erythrocytes, leukocytes, as well as platelets. Okay, that's formed elements. All right, so formed elements and plasma. Plasma is the fluid part. Formed elements are the cells or the fractions of the cells, the fragments of cells. So as I mentioned, one of the main functions of blood is transport. So we transport the various things through the blood, hormones, nutrients, wastes, and gases as well, parts of immune system, and such. So it transports various things throughout our body. So number one function is transport. It has 2 other big important functions, and those are regulation and then protection. Okay, so the regulation part has 3 actual subparts. So when you look at the regulation function of the blood, it can regulate pH or temperature or fluid balance. Okay, the pH regulation has to do with blood containing normally hydrogen and bicarbonate, which are the molecules that are going to be responsible for changes in the pH. But blood also contains, in the plasma part, it also contains buffers. So various molecules that are able to absorb acid or extra acid or base. And we'll talk more about a pH and buffering system when we look at the urinary system. So for now, just know that blood contains buffers, and these buffers prevent spikes in pH. Regular pH of blood is between 7.35 and 7.45. So it's a very narrow range. It's very important that we keep that pH in that small range. If it gets out of that range, it can cause denaturation of proteins, which means you unfold the proteins. And if you change the shape of a protein, it doesn't do its function. So it's very important to keep that pH where it is supposed to be. Okay. It also regulates body temperature. So blood has a regulatory function in terms of body temperature. Blood is going to absorb heat from the processes, the metabolic processes within your body, and then as it absorbs the heat and it travels through your body, it carries that heat to other parts of your body. So technically, your blood is going to warm up your body. And someone just asked, what are the buffers again? Buffers, think about buffers as sponges for extra acid or base. And there are various reasons how acid or base could get into your body. Metabolic processes, from your digestive system, you know, as you digest stuff, some things are acidic, and so that can shift your pH. But these buffers will prevent the change of pH to be large. Okay, so buffers are like sponges for acids or bases. They keep the pH in a narrow range. Hopefully that kind of answered that question. And again, we'll talk about buffers a little more in urinary system, when you'll kind of have a little better understanding of where these acids or bases could come from, because we haven't really talked about that yet. So let's go back to the body temperature and talk about the regulation of the body temperature. Uh-oh, I have two people that are waiting to be admitted here. All right. And okay. So as far as the body temperature, as I've mentioned, the blood, as it passes through our body, it absorbs heat from the metabolically active places, and it takes it to other regions and warms up our body. We'll also see that the way that our body can cool itself is by altering distribution of blood flow. So like during exercise, hold on, let me see. Okay, so someone cannot hear anything, but I know some other people are hearing me, so I know it works. So let's do some tech. All right. Make sure that your device has volume turned on. Like usually most of my devices have volume off, and that might be it.
Tell them to press the mic and then connect to audio.
Oh, okay. So
You might have to type it in since they can't hear.
okay. Hold on. How do I? Oh, okay. Jose just typed it up for me. Thank you, Jose. Oh no, that's fine. I'm trying to like figure out how to actually direct it to the person here in this chat. Yes, it will. So Evelyn just asked if this will be on Blackboard, as you can see in the chat, and it will be. I'm recording this. So if you're already watching this, you'll just kind of skip over this part, right? It at least allows you to catch up on your notes, right? If you're writing as you're listening. So I hope Alejandra got it working. I'll wait for her to, well, hopefully she'll write me back. So anyway, let's go on with the temperature again. And so for body temperature regulation, just know that blood warms up the body, and it also is able to carry extra heat towards the skin if we're trying to cool ourselves. And that's all part of the body temperature regulation. And then blood also controls or regulates fluid balance. And so when we'll talk about the composition of plasma here in a moment, but plasma contains things. It's not just water. It has nutrients dissolved in there, it has electrolytes, it has proteins and various things. And so if you think about blood being in vessels, vessels are surrounded by tissues, right? They run through tissues, and tissues are made of cells and interstitial fluid. And so you will have an area inside of the vessel that is the blood that has a certain concentration of various solutes. And then there is another area on the outside of the vessel of the capillary, specifically capillary is what the vessel type we're talking about, and that's the interstitial fluid. Interstitial fluid also is not just pure water, it has things there too. It has nutrients and various molecules there as well as electrolytes and such. So if you were to pause the scenario for just a moment, you would see that the concentrations would be either the same or different. Okay, if the concentrations are different in the two different compartments, this drives movement of fluid in and out of blood or in and out of the interstitial fluid. Hold on. So that's that. But the proteins in the blood are responsible for what we call colloid osmotic pressure. And colloid osmotic pressure is a pressure. Oops, we got 2 new people. Oh. Colloid osmotic pressure is the pressure that is a result of proteins being inside of the blood. Usually when we talk about colloid osmotic pressure, people want to cry, but it really is not that difficult to understand. And it will make a little more sense when we talk about capillary exchange in chapter 20. So colloid osmotic pressure is the pressure of fluid moving in the direction of high concentration of proteins. Okay, so we will actually see that blood plasma contains various proteins. Those are a regular part of blood plasma. And interstitial fluid has some proteins, but they're not, it's not that many as in the blood. And so there's a constant pull of fluid from the interstitial space into the blood because of these proteins that are in the blood. So think about proteins as being magnets for water, okay? And since there's always more proteins in the blood, there's always going to be this pull on fluid from the interstitial space into the blood. Now, when we talk about capillary exchange, we are going to talk about other pressures there as well. So we're just looking at this one. But just in general, a concentration of solutes in the blood is going to determine which way fluid moves between the compartments in your body, okay? And so that's how the regulation of fluid balance works, kind of in a nutshell. And then the third part of the blood function is protection. So blood there, and this is kind of a twofold thing. So the blood contains leukocytes. These are formed elements. These are your white blood cells, and leukocytes are cells that are going to be used to fight infection, fight pathogens, and you know more about those in chapter 21. So we have these cells that are part of our immune system, but also the blood plasma contains proteins that are called antibodies. And antibodies are molecules, these are protein molecules that are designed specifically against a certain type of pathogen. And so there's a molecular level in protection as well as a cellular level of protection. So those are the functions of the blood, okay? Transport, regulation, protection, okay? 3 main things. As far as characteristics of blood, the color of the blood is red, and it's a different shade of red depending on whether the blood is oxygenated or deoxygenated. Oxygenated blood is is fairly bright. It's a bright red color. Deoxygenated blood is going to be darker in color. Blood is not blue. If you had a look at chapter 18, it kind of explains why, you know, you might look at your vessels, right? And you see, but they kind of look blue and some look purple. And the reason for that, and the reason why you're seeing this is because your skin kind of acts as a filter for some of those shorter wavelength colors. And so you're only seeing the longer wavelengths of colors, which is the blue. But if you were to actually dissect an arm, it wouldn't be blue, okay? So blood is not blue. We all have red blood, okay? A volume of the blood is about 5 liters on average. The volume of blood in males is slightly higher than in females on average. And that's just because female body is on average smaller than an average male body, okay? So they would have less blood than males. And viscosity of the blood refers to thickness. Thickness of blood has to be thicker than water, right? And that's because it is water plus other things, okay? And so it's about 4 to 5 times thicker than water, okay? So blood is about 4 to 5 times thicker than water. This changes because composition of blood is constantly changing. Your blood composition is never the same. It fluctuates all the time because things move in and out. And so viscosity depends on the amount of fluid that you have in your blood. The more fluid you have in the plasma, the less viscous or less thick your blood would be. The plasma concentration is referring to concentration of various things that are found in your plasma. So again, this kind of has to do with viscosity also. You know, the concentration is going to be lower the more fluid you have, and vice versa. The concentration increases if the fluid decreases or solutes increase. So, and again, this plasma concentration is a drive for fluid moving in and out of the different compartments. So that's a plasma concentration. Temperature, we already know that blood regulates temperature, but I forgot to mention to you what the actual normal temperature is for blood. The normal blood temperature is about 1 degree Celsius higher than the measured body temperature, okay? And so that's how I was saying that the blood warms your body as it's passing through your body, okay? So blood temperature, like if we're measuring core blood temperature, is about 1 degree Celsius higher than what you measure as your body temperature, okay? I've already mentioned pH. pH is supposed to be within that range of 7.35 and 7.45, that's basically your functions and characteristics of blood, kind of an introduction here. What we're going to look at now is we're going to look at this, okay? So I'm sharing with you a figure from McGraw-Hill, and now this is showing my Zoom window really weirdly. So if you, where's the chat? I don't know if it actually shows me the chat here. Okay, here it is. All right, so I gotta close this. All right, so let's talk about this picture. And this image, you can kind of figure out a lot of things from this screen right here from this image. So what I would like you to know before the exam is that, what is the composition of the whole blood? This test tube here in this image shows you what blood would look like if you put a test tube of whole blood into a centrifuge. Centrifuge is an instrument that spins really, really quickly. As it spins, components of that test tube would be separated into different layers based on their weight, okay? And this is what the whole blood would look like after you spin it, after you centrifuge it. The most of the whole blood is what? It's plasma, right? So you can see that 55% of blood is plasma. The erythrocytes, or the red blood cells, are a little less than half, okay, about 44% or so. And then the middle layer there, about 1% or less sometimes, is going to be the buffy coat. Buffy coat is where you find the leukocytes and your platelets. Okay, so let's go ahead and begin with the plasma part here. We'll start from the top and talk about the specific components of the plasma. So you can see that that plasma is mostly water. It's 92 to 95% water by weight. Okay, most of the plasma is water. The other component of plasma would be proteins. And when we look at the group of proteins that we see here, we see that the most abundant protein is or are albumins. Okay, these are your most abundant proteins. Albumins are transport proteins. Albumins are small transport proteins. Globulins are also transport proteins, but they're large transport proteins. Okay, I was wondering if I would see if somebody chats. And I do see it. The question is here, do we need to know the exact percent for our exam? I would like you to know the percent of the whole blood. So like the 55, 1, and 44. I am not going to ask you percents for albumins or globulins, but I would like you to know that the most abundant proteins are albumins. Okay, so you don't have to tell me it's 58%. Okay, but know that it's most abundant proteins are albumins. Also know that most of the plasma is water. Again, you don't have to tell me it's 92%, but no, that's the most, that's the largest component of plasma is water. Okay, so albumins and globulins are transport proteins. Remember those lipid-soluble hormones from chapter 17, and how we said that they require a carrier protein? Albumins would be the most likely ones to be the carrier protein for these hormones, or globulins too. They're also transport proteins. Another protein worth mentioning, even though it's not a very big percentage of those plasma proteins, it is worth mentioning, however, and that's the fibrinogen. Fibrinogen we'll mention at the end of this chapter. It is a protein that's involved in blood clotting, and fibrinogen in this fibrinogen state is actually soluble, but during blood clotting, there are pathways that will convert fibrinogen into fibrin, and when it becomes fibrin, it's called fibrin because it looks like fibers, and it's going to provide a framework for the blood clot, so that it's going to hold it all together. And then there are some other regulatory proteins that are in a very small abundance in the plasma as well. These are the main ones here. And then we have other solutes also in there, such as electrolytes, nutrients, waste products, and gases. This is about 1% by weight. They're very small molecules, so it's not a big part by weight of the plasma, but it's a very important thing, right? Because that's how nutrients and electrolytes and everything travels through the blood. So that is our plasma. Let's go ahead and talk about this blue part, this blue box. It's funny when you look at this picture. The buffy coat is the smallest part of the whole blood, but it gets the biggest box. Okay, so buffy coat is the part of, and by the way, buffy coat and erythrocytes are formed elements, as you can see here, right? Okay, so these are your formed elements. So we can split formed elements into the two parts, buffy coat and erythrocytes. Buffy coat contains platelets, and it also contains leukocytes. Platelets are fragments of cells. They're actually made from a cell that we call megakaryocyte, and they're just like pieces of cell membrane with some cytoplasm inside, and then there are some proteins there as well. But they're formed from these megakaryocytes. And I'll show you actually the pathway of how this is done when we talk about hemopoiesis. But platelets are important in blood clotting. For proper blood clotting and the formation of blood clot, you have to have platelets, and you have to have a good number of platelets. Okay, if you're low on platelets, if your platelet count is low, then that could affect your blood clotting. Leukocytes are your white blood cells, and we have five main types here that are described. And actually, lymphocytes that you see there in the immune system chapter will actually divide lymphocytes and talk about like specific types of lymphocytes later. But here they're just grouped together as lymphocytes. For leukocytes, I would like you to know what's the most abundant leukocyte, and that's your neutrophil. Okay, half to 70% of all your leukocytes are neutrophils. And then again, you don't have to know the percentages, just know the most abundant leukocyte is neutrophil. Then we have some other ones here. We have monocytes and lymphocytes, basophils and eosinophils. If you look at this picture closely, okay, look at that box right here with the leukocytes. You can see that neutrophil, and it's not super obvious until you like really look at that neutrophil there, but you can kind of see that neutrophil is dotted, right? It has like little kind of pink dots inside. Eosinophil is really dotted, right? It's got spots, red spots on it, and basophil has blue spots on it, okay? This refers to the way they actually stain, but these three cells, neutrophils, eosinophils, and basophils, are known together as granulocytes because they have granules. So those spots are actually granules. And so they're called granulocytes. The other two, the monocytes and lymphocytes, these do not contain granules inside of their cytoplasm, and so they are called agranulocytes. The letter A at the beginning of the word means that it's without, okay? So agranulocytes. So granulocytes, agranulocytes. Just a brief introduction about what they do. I'd like you to know briefly what a function of each of these is. So your neutrophils. Neutrophils are, again, our most abundant leukocytes. These are usually first ones to arrive at a site of infection. Neutrophils also are going to be engulfing bacteria preferably, okay? So neutrophils really prefer bacteria. They like to eat bacteria, okay? That's the main part about neutrophils. The eosinophils, these are good, like their characteristic feature is being good at fighting multicellular parasites. So like worms and such. I can't think of anything. Well, yeah, most commonly it's worms for the multicellular parasites. Basophils are important in allergic reaction. Okay. The basophils are going to release histamine and heparin. Histamine is a vasodilator. Heparin prevents blood clotting, and they're going to be also really important in inflammation. Okay, again, more of that will be kind of clear when we get into the immune system chapter. Okay, immunity.
Okay, you said they release histamine and what else?
Histamine and heparin.
Okay, thank you.
You're welcome. So histamine, again, it's a vasodilator. So if you have allergies and your eyes are all watery and your nose is all runny, it's all that vasodilation, all this fluid is now allowed to kind of leak out of your membranes there, okay? And so you would take antihistamine to fight that, right? And then heparin prevents blood clotting. So it allows blood flow to go through so that repair can happen more efficiently, okay? And so that's basophils. Lymphocytes, there are 3 types of lymphocytes, and depending on their type, their function is different. So we have T lymphocytes. T lymphocytes are going to be mostly regulators of the immune response. We have B lymphocytes. B lymphocytes are going to produce antibodies. And then we have natural killer cells. And natural killer cells, as their name suggests, are going to be good at killing things. Okay, they usually kill cells, is what they do. So that's lymphocytes. And then the last one here in the center, the biggest cell that we have in that box is monocytes. And monocytes are cells that will ultimately become macrophages. And macrophage is a cell that likes to eat, and it likes to eat anything. It doesn't, it can eat or engulf infected cells. It can engulf debris in an area. It can engulf viruses, bacteria. It doesn't really care. If it's not right, it's not supposed to be there. Macrophage will come and get it and eat it. It's about as selective in eating as I have been through this quarantine. I eat everything too. So that's macrophages. Monocytes become macrophages. And that's our buffy coat. And then the last part that we see here are erythrocytes. And erythrocytes are, okay, hold on, let me see the chat. Somebody has a question. So leukocytes, neutrophils, lymphocytes are all cells, right? Yes, that's correct. So this part, okay, these are cells of your buffy coat. Notice I didn't include the platelets there because platelets came from a cell, but these are just fragments of cells. And then erythrocytes, erythrocytes are funny because I was gonna say erythrocytes are also cells, but technically they're not really true cells, and you will see why here in a moment. Okay, but yes, the neutrophils, eosinophils, basophils, monocytes, and lymphocytes are cells, true cells with a nucleus and all. White blood cells is what they are. And the phils, see the three that end with phil, neutrophil, eosinophil, basophil? The three phils are your granulocytes, okay? The other that don't have the phil in them, those are not granulocytes, those are agranulocytes. Okay, so let's look at erythrocytes. And erythrocytes are, as when you look at the part that they make up in the whole blood, they're not the biggest component of the whole blood. It's only about 44% or so. And in males, actually, that percentage is a little higher. But erythrocytes are the most abundant formed element. Okay, so I'm going to repeat that. They're most abundant formed element. Okay, what that means is that out of these two boxes, they're the most abundant ones. Okay, so you don't have to remember the number here, okay, but when you look at that and compare it to the other, I mean, it's even visually obvious, okay, when you think about formed elements being just this part of that test tube, erythrocytes are the biggest part, right, of the formed elements. Okay, the erythrocytes are red blood cells, and we call them red blood cells, but technically that is actually kind of incorrect because the erythrocytes are not really a true cell when they're in their final stage. And this is because the erythrocyte in a final stage does not have any organelles inside. So in the process of erythrocyte formation, we lose nucleus, we lose all the organelles from those cells. All you are left with is this cell membrane with this biconcave shape and some proteins that are holding that shape, and then hemoglobin, bunch of hemoglobin in the cytoplasm. That is an erythrocyte. So because it doesn't have organelles, technically we really should not call it a cell, but we do still. So that's your erythrocytes. And again, erythrocytes are about 44% of the whole blood. So I want to look at, this is what the blood would look like under the microscope. So notice that erythrocytes are smaller than your leukocytes, right? Erythrocytes are these, all of these right here. And you can kind of see how in the middle, they look like they're clear. That's because that's where they're thinnest, okay? So Those are erythrocytes. And then you have here and there, you have some white blood cells. And you can see that white blood cells are larger. They also stain darker, way darker than the erythrocytes. And then it's kind of hard to see on this image here, but I guess they did find a little group of platelets here. So right here, okay, that's a group of platelets. If you actually look at the histology picture, okay, where did I put this? Okay, and I hope you are seeing my blackboard right now, but this is being slow. Okay, so here in the unit 1, as I mentioned, is, oh no, I put this in documents. So. Okay, there's this histology file. And if you have watched the video that I attached to that endocrine lab handout, I have mentioned that I was going to upload histology file for you. Okay, you know what? This is trying to download it and it's killing my connection. But in that file, you can see a picture of blood. I have a blood, and that's the one, like, I took most of those pictures. Some of those pictures were from students, but they're from our slides in our lab. That's except for the first one. So on that one, you can see a lot of platelets all throughout in that file. And they're a little tiny. I mean, notice how small they are. Oops. Okay. See how small they are compared to the others, like compared to the actual cells. Those platelets are super, super tiny. Again, a picture in that histology file has more platelets. They're way more visible. About the same color still. But yeah, so that's that. This just kind of summarizes some information for you in a table format if you want to look at that. You don't need to memorize that, okay? Don't. So you can skip this table and this one and this one. Tables are great for reference for the most part, okay? Especially if we're talking about numbers. But this is the picture that I wanted to get to. So what I want to talk about now is hemopoiesis. And hemopoiesis is formation of formed elements. So this is how we form formed elements. So it looks like there's a lot of different names on here. There's a lot of different stages, a lot of different chemicals involved. So I'm going to try to simplify this for you, okay? I'm going to tell you what I would like you to know. So I would like you to know that what hemopoiesis is, okay? So make sure that you know the definition of hemopoiesis. Oops, that's not highlighting very well. Okay, maybe I will not use highlighter. Okay, so make sure you know what hemopoiesis is. Then I want you to know that hemopoiesis is formation of formed elements, and it begins with a cell known as hemocytoblast. Okay, hemo for blood, cyto cell, and blast is like a precursor to something. Okay, it's a cell that will become something else. All right. So hemocytoblast, know that this is a blood stem cell. Now that blood stem cell can be stimulated to go through one of two processes. Okay. And we're not getting into what stimulates it. We're not getting into what, who that messenger actually is or how chemically does this happen. It's just know that sometimes the hemocytoblast will go through the lymphoid line, and sometimes that hemocytoblast will go through myeloid line. Okay, line just in this process, or in this term or this sense, it just means a pathway. Okay, that's the pathway that it takes. So in the lymphoid line, I'm going to start from the lymphoid line here. Okay, the lymphoid line will go through the process of leukopoiesis. Okay, so know that. Okay, hold on, somebody came. Oh, And all right. All right, so the lymphoid line, okay, will go through, it can only go into the process of leukapheresis, okay? No other process is possible from the lymphoid line. It will only go through leukapheresis, okay? And I kind of wrote over that, but it's this process right here, okay? So lymphoid line goes through the leukapheresis and it forms one of these three cells right here. So it forms B lymphocytes, T lymphocytes, and natural killer cells, all right? So lymphoid line gives you B cells, T cells, and natural killer cells through the process of leukapheresis. All right, that's what I want you to know about the lymphoid line. Since we're talking about leukapheresis, looking at this table, or this flow chart, you see that leukapheresis can also happen from the other line, from the myeloid line, okay? So from the myeloid line, we can get leukapheresis as well, okay? And the leukapheresis from myeloid line gives you these guys. All right, so it gives you eosinophils, basophils. So it gives you the granulocytes and the monocyte is leukapheresis from myeloid line. All right. Notice I skipped all those intermediate names there, so I'm not gonna ask you to like fill those in there, at least not for leukapheresis. Just know what leukapheresis from lymphoid line gives you and what does the leukapheresis from myeloid line gives you, and you'll be good. There are these white kind of like tags here with some letters in there, and these are to signify the stimulating factors that are determining what that cell will become. Okay, so there are like chemicals, and if you have granulocyte stimulating, colony stimulating factor, then it will create granulocytes. I'm not going to make you match those to the types of cells, so just know that those are the things that affect formation of the final cells there. Okay, so that's our leukapheresis. There are two other processes in the process of hematopoiesis, and they are erythropoiesis and thrombopoiesis. Okay, I'm gonna, since I started on the right, I'm gonna move just kind of in order to the left here. And so let's go ahead and look at thrombopoiesis. Thrombopoiesis is a formation of platelets. And this one, I do actually kind of want you to know the name of this cell right here, and that's megakaryocyte. Okay, so thrombopoiesis involves first formation of megakaryocyte, and then the megakaryocyte gives rise to the platelets. And the way that this works is something like this. All right. This image right here shows you how those platelets are formed. So you have your big, large cell called megakaryocyte. Okay, this megakaryocyte has all these like pieces of cell membrane kind of sticking out of it, and we call those proplatelets. Okay, so all of these, like see this and this and this, and these things that hang off of it, those are the proplatelets. So megakaryocyte will sit on the outside of the capillary. Okay, so this is a blood capillary, this blood vessel right here. And we can see that blood flow goes this way, right? And so as the blood flows, a megakaryocyte sits on the outside of that capillary, and the proplatelets stick between the cells of the capillary to the inside of the capillary vessel. The blood flow will slowly kind of just chop off pieces of those proplatelets. Okay, they will get pieced off. So the flow of that fluid, flow of that blood will cause these proplatelets to break off in pieces. And that's what creates your platelets. So eventually, as we chop off more and more platelets off of this megakaryocyte, it does get smaller here. And then eventually, when it's all gone, macrophage would be a good one to come and eliminate it, right? So That's how thrombopoiesis works. Again, thrombopoiesis is formation of platelets, okay? And it involves that megakaryocyte. Mega because it's really big, okay? And then the last process here is erythropoiesis. Erythropoiesis is formation of erythrocytes, okay? Now for this one, I actually do want you to know this EPO thingy here. EPO stands for erythropoietin. Erythropoietin comes from the kidneys and it stimulates formation of erythrocytes, okay? Erythropoietin is kind of interesting because it comes from kidneys and it stimulates formation of erythrocytes. But it's also interesting because of what stimulates erythropoietin. So what stimulates erythropoietin release from the kidneys are low oxygen conditions, as well as testosterone has an effect on this. So if you're in places, if your breathing is impaired, that would be low oxygen for your body, right? Or if you're in a higher altitude, where oxygen levels are lower, that would stimulate your kidneys to create more erythrocytes. So like, well, stimulate the erythropoietin release so that you can create more erythrocytes. Because more erythrocytes means more efficient delivery of the oxygen throughout your body, okay? And then testosterone also has an effect on this. And this is also why, one reason why the percentage of erythrocytes in a male body is usually a couple % higher than in a female because of that testosterone. So anyway, so erythropoietin stimulates formation of erythrocytes. What you see as this erythrocyte is being created, you see that the cell is getting more and more red. Okay, the names are changing also, but the cell is changing color. And the color change that you see here is due to formation of a protein that we call hemoglobin. Hemoglobin is your main player in carrying oxygen. Hemoglobin is what binds oxygen. Okay. And I'll talk about structure of the hemoglobin a little later. We can also see right here, towards the end, nucleus is being ejected. So we kick out the nucleus, we also kick out all the other organelles, and what you're left with is this reticulocyte. Okay, reticulocyte will enter the bloodstream and then it will take it a day or two to mature in order to become your full-blown erythrocyte. Okay. Now, because erythrocytes do not have nucleus, and really all you are left with is just this plasma membrane with cytoplasm inside and hemoglobin, erythrocytes have a definite lifespan, and they live about 120 days, and then your body recycles them. Your body recycles erythrocytes in liver or in spleen. Liver is kind of the most common one, but spleen also recycles erythrocytes. So we break them down. Let's look at the structure of the hemoglobin here somewhere, okay? So hemoglobin looks like this, okay, there we go. And it's a protein that's made out of 4 chains. It has 2 alpha chains and 2 beta chains, globin chains. And each chain inside of the chain has one heme group with a porphyrin ring. It looks a little bit like a Saturn there, right? It looks like the planet Saturn with the ring there. The heme group in the center, okay, that red dot is supposed to symbolize or represent iron atom. An iron atom is what binds oxygen, okay? So each hemoglobin has 4 globin chains. Each chain has 1 heme group with the iron atom inside. So how many oxygen molecules can each hemoglobin carry? Well, I kind of circled them for you already right there. And so you see that there is 4 different irons on each hemoglobin molecule. And so each hemoglobin molecule can carry 4 oxygens. All right. I just saw this chat right here. It didn't ding at me for some reason. So I'm not really sure what you wanted me to zoom in. Is it this one or the last one? And yeah, sometimes, hold on. Can I zoom in here? Oh, okay. Whenever I put it into this software, the pictures, they also get a little fuzzier. I don't know why they don't retain their original quality. So if you are somewhere where you can have your textbook with you, you might want to find that figure in your textbook also. Yeah, because the last figure was kind of big and had a very small letters. So I'm sorry I didn't see your message earlier. Okay, so let's look at the hemoglobin again, just to kind of wrap the hemoglobin up. Hemoglobin is a protein that is composed of 4 globin chains. It has 2 alpha chains and 2 beta chains. And each chain has a heme group. So I'm going to circle them again. So here's one for this chain. Here's another one. Here's another one. And here's another one. Okay, so each globin chain has this one heme group, and in the center of this heme group is an iron atom. So this is that Fe that you see here. Okay, hold on. Let me see if I can zoom this. Oh, look, I can make this bigger. I hope it gets bigger for you too. It got bigger for me here. And so the iron is what binds oxygen. And since each hemoglobin has 4 heme groups and therefore 4 irons, each hemoglobin can carry 4 oxygen molecules. Okay, so that's our hemoglobin. The hemoglobin molecule does not only carry oxygen though. Okay, a lot of people think that that's the only way that oxygen moves throughout our body, and hemoglobin is only for oxygen, but hemoglobin is not only for oxygen, it is also for transport of small percentage of carbon dioxide, and hemoglobin can also bind hydrogen ion, believe it or not. So those two, the carbon dioxide and hydrogen ion, they don't bind to the iron. They bind to the chain somewhere, like, you know, I don't know, maybe right there or somewhere. But yeah, it can carry other molecules also, specifically carbon dioxide and hydrogen. All right, let's go back to the normal size. And yeah, so we know that hemoglobin or erythrocytes have a definite lifespan, so about 120 days, and then they get recycled. When we recycle the erythrocytes, we then release all these hemoglobin. So we also have to recycle hemoglobin. So our bodies do recycle hemoglobin. We break the hemoglobin down into various things. Globins, those globin chains, they're just proteins. So we chop them up into amino acids, and then we can use amino acids to build other proteins. I always think about amino acids as Lego pieces, right? You know, you can always disassemble the protein and then you can reassemble it into something else while, you know, keeping those basic building blocks, right? The iron, a lot of iron gets actually reused. So we recycle a lot of the iron that comes from hemoglobin. Some iron is lost in urine and feces, as well as sweat. And then the heme group, okay, this thing, the heme group is broken. Wow, it's really delayed as I'm writing on this. The heme group has to get broken down chemically into smaller things, and it gets broken. Ultimately, it gets broken down into bilirubin and biliverdin, and then those get eliminated out of your body. So that's the breakdown of hemoglobin. What else about hemoglobin and erythrocytes? Here is a picture of erythrocytes. They look like an incomplete donut, or they have a you know, they're fairly narrow on the inside, like, you know, like a squished incomplete donut. All right. This figure right here shows us the release of erythropoietin. So it tells us what the stimulus is, and then, you know, kidneys release this erythropoietin, and then that stimulates formation of erythrocytes, stimulation of, erythrocyte formation. Hemopoiesis happens in the red bone marrow. I don't think I mentioned that, but that's the site of hemopoiesis in adults. Like during fetal development or embryonic development, hemopoiesis cannot happen in the red bone marrow because there's no red bone marrow, right? And so for that reason, and during embryonic development, hemopoiesis happens in the yolk sac. So this is where your first erythrocytes and blood vessels get formed. Then as the embryo grows into fetus and fetus continues to grow, then hemopoiesis happens in the spleen and liver. And then ultimately as bones form and red bone marrow forms, then the process starts happening there as well. So that's hemopoiesis. And formed elements. So I don't like to keep us over an hour, and it's been, even though we started a little later, it has been over an hour. The only thing that is left for this is hemostasis and then blood typing. So I will make a recording. Actually, I think I might make two so that you can see blood typing in one, and I'll talk about the lab in there a little more also. And then I'll make one on blood clotting or hemostasis. That way you're not watching like a really long video. Okay. So I'm gonna go ahead and stop with chapter 18 here, and then the rest again is going to be on another recording. Okay. So any questions, anybody? I think I have sound, yeah. Or let me see if anybody's chatting. Okay. Yes, test 1 is chapter 17 and chapter 18. And it's going to be, I don't know if that might be a follow-up question, but it will have questions from the lectures, from the textbook. You know, some might resemble some Learn Smart questions, but it will also have things from Visible Bodies, where I'll get a visible body and I will like save a picture of a structure on that visible body and have you tell me what it is. I can't make it clickable like in the Visible Body. I like that, where it gives you like the practice with the anatomy idea and it tells you click on this, and then but I can't make questions like that. Mine have to be still in on the exams, but that's okay. What were the 3 cells of the lymph? Okay, so for lymphocytes the 3 cells for lymphocytes are T lymphocytes, B lymphocytes, and natural killer cells. And yes, you are supposed to be working on the labs also. So as you're going through this, I mean, everything, as you were reading the chapter 17, again, the endocrine, I think it's a Word document, isn't it? The endocrine lab exercise, it really supplements your reading, especially like when you look at those tables, you're putting all those hormones there together, in one place. And then I'm asking you to draw some of those pictures. And then with Visible Body, you're practicing structures, what is what, but also the Visible Body has some, like a little shorter explanation of things, but it's got things that the textbook has too. So you're looking at things multiple times, and that's what makes you remember things better, right? So I think I forgot the due date for the endocrine sheet, okay, where you're supposed to fill in all those hormones, and I think that might be today. So check the due date so that I can look at that and give you feedback before the exam. Oh, tomorrow, okay. Okay, so somebody's talking about the quiz and on the visible body, and sometimes mine is fairly easy whenever I'm playing around with it. But if you double click on the region that you want to zoom into, it will nicely zoom into that area, and then all you have to do is rotate for some of those, as opposed to just looking at the whole body and then trying to zoom into that certain area. I saw that. I mean, I found out that double clicking on something in the region where you're trying to look will zoom in well for you. So hopefully I mean, this is what, day 3 that we're using it, so you'll get better at it as we're going along. Okay, you can't be a pro at it or a master at it in 3 days, so it's all about habit. So anything else? Okay, I don't know, because this doesn't show me if anybody is typing anything, but you know that anytime you have a question about anything, you can email me. I try to get back to you as quickly as I can, as quickly as possible. So anytime you need help with anything, let me know. I'm here almost all the time on this computer, so all right, so let's go ahead and end this. Oh, you're welcome. Thank you guys for coming today, and I'll record the other parts of the blood either today or tomorrow, so you have them. All righty, you have a good day.